All right, good morning, everybody, to another ShareWise webinar. Today, I have the privilege of being joined by Kilian. Kilian's the CEO at Cynata Therapeutics, and I just wanted to introduce yourself, Kilian, and also if you could just introduce and give us a bit of a general overview of Cynata and what you guys are up to at the moment. And then, as I believe you've got a presentation you'd love to take us through.
Yeah, that's right. Thanks, Luke, and hi to everybody. Thanks for joining. As Luke said, my name is Kilian Kelly. I'm the CEO and Managing Director at Cynata Therapeutics Limited. We're an ASX listed biotech company based in Melbourne, and we're focused on the development of stem cell and regenerative medicine products for a range of serious medical disorders. And obviously, I can elaborate on that in my presentation.
Yeah, beautiful, mate. Well, I'll leave the floor up to yourself, and then if you wanted to share your screen, take us through the presentation. And I'm happy to ask. I've got a few questions I would be keen to ask you at the end of the presentation. And then for anyone that wants to ask a question, feel free to put it in the chat, and I'll come to that at the end of the presentation as well. But the floor is yours, Killian. Thank you.
Okay, sounds great. Thank you. So, as I said, Sonata is an ASX listed company with the ticker CYP. We are based in Melbourne, and we're focused on the development of cell therapy products, a specific type of cell therapy product, which we call MSCs or mesenchymal stromal cells, also known as mesenchymal stem cells. All of those terms mean the same thing. And some of you may have heard of MSCs from other companies and indeed academic institutions, as they've attracted a lot of interest over the last 20 years or so, in particular, to treat a range of serious medical disorders. But what's different about Cynata is how we make these products. So we have a unique platform to make MSCs from a single blood donation once and at scale. And again, I'll explain more about that in a moment. Now, for those of you who are new to MSCs, you might be wondering what they are and why we would be interested in them. Well, basically, MSCs are cells that occur naturally in the body in small numbers, but they are of interest because they have the ability to balance the immune system, reduce inflammation, and promote tissue repair and regeneration. But a big bottleneck that has held the field back has been how to make these cells consistently and at scale, and that's what we solve using our unique platform. We have 4 active clinical programs right now, including phase 2 and phase 3 trials, which are nearing completion. I'll come back to that in a moment. First of all, a bit of an overview of the company. As I said, we are ASX listed. We have a market cap at the moment of just over $60 million. We have sufficient cash in the bank to give us runway into the second half of next calendar year or the start of next financial year. On the register, we have some relatively large holders, including Bioscience Managers, Fidelity International, and Fujifilm. People might be curious about that one. Fujifilm is a major player in the manufacture of cell-based therapies, and we have a strategic manufacturing partnership with them. Overall, the top 20 actually own about 45% of the shares on the register, so it really is tightly held. Now, talking about those clinical programs again, what's really getting everybody excited at the moment in particular is our osteoarthritis and graft versus host disease trials. The osteoarthritis trial is a phase 3 trial, which has now completed in terms of patient visits, and the results are being analysed as we speak. So we expect to be able to announce results of that trial in Q2 next calendar year. And similarly, the graft versus host disease trial, which is a phase 2 trial ongoing right now, we also expect to have results of that in Q2 next calendar year. So it really is a hugely exciting time for the company with 2 enormous inflection points coming up. The other clinical programs that we have going include a phase 1/2 trial in kidney transplant patients, and we very recently completed the first cohort of 3 patients in that trial. And last week we announced that we'd had a DSMB or a Data Safety Monitoring Board review of that first cohort, and everything was in order, and we're now progressing towards cohort 2. And finally, we have a diabetic foot ulcer program where late last year we completed a phase 1 clinical trial in patients with diabetic foot ulcers, which showed very promising results. So a little bit more about MSCs. As I mentioned, these cells do occur naturally in the body, but they occur in very small numbers. And the idea of MSC-based therapy is to produce larger numbers of these cells and then administer them to patients, much like you would a normal pharmaceutical product. The first use of MSCs in a human patient was in 2004 in Sweden, when a 9-year-old boy had advanced graft versus host disease. He had exhausted all treatment options and he was not expected to survive. He was then treated with MSCs and became the first person ever to be treated with MSCs. And he made a complete recovery. So, of course, that was an amazing result, and it really caused an explosion of interest in the use of these cells to treat various diseases. Since then, there's been more than 1,800 clinical trials started around the world with these cells. They've been conducted by a whole range of different companies, universities, and hospitals. And so we're now at a stage where we're really entering a period of maturity where these products are heading towards commercialization. And that really brings into focus the need to be able to make these cells in a scalable way. And as I mentioned at the start, that has been a real challenge. And the reason is that conventional means of making these cells rely on tissue donors. So, for example, they need people to donate bone marrow, and they then isolate MSCs from the donated bone marrow. And what I mean by that is bone marrow contains lots of different types of cells, but most of those cells are not MSCs. So they have to isolate the MSCs, which is relatively straightforward, but it yields a really small number of MSCs. As an example, maybe about 20,000 MSCs, which might sound like a lot, but it isn't, because you need usually hundreds of millions of cells to treat just one patient. So, of course, if you need to treat many patients, you need billions or even trillions of cells. So in order to turn 20,000 MSCs into that sort of larger number, they have to grow the cells in the lab. That's a process known as culture expansion. And that works to an extent in that you can make quite a large number of cells, but there's a limit because they stop growing eventually, they stop reproducing themselves. And even before they get to that limit, they start to lose functionality and potency. So what this means is there's a limit to how many doses you can make from one donor. It might be something like a few thousand doses per donor, which at commercial scale really isn't very much. So they keep needing to go back and getting new donors all the time. Now that's a logistical challenge, it's costly. But more importantly, there's variability between donors. So every time a new donor provides bone marrow, they have to get MSCs, which will be a bit different to the previous donors. And that means when you have this inconsistency in starting material, you're likely to have an inconsistent end product. So that's a real fundamental challenge, which is quite a major problem when you're trying to make a pharmaceutical-like product, which is expected to be consistent time and time again. And so that's what our platform is intended to address. So we use a completely different approach to make our product. We have a technology based on what is called iPSCs, or induced pluripotent stem cells. Now, I won't go into too much detail about what iPSCs are, but they are basically cells that come from an adult donation, in our case, a blood donation, and they're then reprogrammed to become iPSCs. And the advantages of iPSCs are that they have an ability to keep reproducing themselves essentially indefinitely. So once you have an iPSC bank, which we do, we can continue to make more of these cells essentially for as long as we want to make our product. We can then use our patented process to turn iPSCs into MSCs. So instead of harvesting MSCs from donors and so on, we simply take a vial of iPSCs out of the freezer, and we then use our proprietary process to turn those iPSCs into MSCs. This then becomes a very consistent way to make the product because we're using the same starting material every time. But in addition to that, it's very, very scalable because, as I said, we can continue to make more and more of these iPSCs as we need them. So ultimately, this is a way to make consistent and potent MSCs time and time again. Now, just a quick word about other MSC companies that people may be familiar with in the ASX. One, probably the most notable, is Mesoblast, or MSB, which is the leader of MSC-based therapies worldwide, in fact. So it's amazing that we have such a company here in Australia. They recently got a US FDA approval for one of their products, which is the first commercial approval for an MSC-based therapy. And that's fantastic news for the whole field. Uh, but one thing that I'd just like to draw people's attention to is just the huge gap between our market cap and theirs. It's about 60 times or thereabouts at the moment. Now, of course, Mesoblast is further advanced. They've got a product in the market. They've got other phase 3 programs and so on. But what that really shows is the opportunity that we have, if we're successful, to significantly grow the value of this company. I should just mention as well that when we make MSCs using these different approaches, as I said, everybody else uses what we would call a donor-derived process, whereas we have our unique iPSC-based process. That actually makes a difference, not just in the scalability of the process, but also in the functionality and the quality of the cells. And we've got lots of data, including this paper, which was published earlier this year, which really illustrates that MSCs made using our process actually have some different properties to donor-derived MSCs. And in various ways, they have superior properties. So that gives us a great deal of optimism that these cells actually work well as being cells that we can make very scalably. Now, moving on to our clinical programs, as I mentioned, the osteoarthritis trial that we're running at the moment is a phase 3 clinical trial, which is taking place in Australia. Osteoarthritis is a chronic joint disease where the cartilage in a joint gets worn away over time. It's very common, especially as people get older. And in fact, I've heard it said that almost everybody will develop osteoarthritis if they live long enough. So osteoarthritis affects something like 600 million people around the world, and it causes huge problems, primarily because there are no existing treatments that change the course of this disease. So all existing treatments treat the symptoms, but they don't stop the actual cartilage degeneration. So the patients keep getting worse over time. And what that means is ultimately they often need knee replacement surgery. Now, that's clearly not something anybody would really want if they had a choice. It can certainly bring a lot of benefit, but it costs a lot of money. There's risks associated with any surgery. And of course, it takes quite a while to recover from. So what everybody has been searching for is a way to actually change the course of this disease and stop the cartilage wearing away over time. And that's what our trial is designed to do. We're conducting this in partnership with the University of Sydney, and it's funded using an NH&MRC grant, which comes from the Australian government. So Cynata's only cost here was to supply the cells that are being used in the trial, which is great news for the company and for our shareholders. But we do still have the commercial rights to use this data. Now, this trial is a large trial. It's a 321 patient randomized controlled trial, and it has two primary endpoints. One is to show the effect on pain and symptoms, and the other one is to assess cartilage thickness as measured by MRI. So this trial is designed to show that disease modifying effect. And if it's successful, this could be the first ever treatment that is actually shown to modify the course of osteoarthritis. And if that's the case, then needless to say, that would be absolutely huge, not just for Cynata, but also for the treatment of these patients. And as I said, final results from this trial are not far away now, expected in Q2 in coming calendar year. Switching tack to graft versus host disease. This is probably a condition that people will be less familiar with. In fact, many people may not have heard of it at all, which is a good thing because it's a very nasty disease, but thankfully it is quite rare. It's a condition that occurs in people who have a bone marrow transplant or similar procedure. And those procedures are usually used to treat blood cancers like leukemia and lymphoma. And they can be very effective, but a big downside is they can cause this condition called graft versus host disease. So what happens is that immune cells in the transplant recognize the recipient as foreign, and it's then like they have somebody else's immune system inside them, recognizing all of their own tissues and organs as foreign. Needless to say, that causes really terrible symptoms, and unfortunately, it's very difficult to treat. The first line treatment is steroids, but they only work in roughly 50% of people. And the remainder of people are known as steroid resistant patients, and they have a very poor outlook, unfortunately, at the moment, with 2-year survival rates of less than 20%. We conducted our first phase 1 clinical trial in these patients, so patients who had already failed to respond to steroids, and we saw really, really encouraging results. So we saw that 13 out of the 15 patients we treated, or 87%, improved by at least 1 grade. That's known as an overall response, and that is the FDA's preferred approvable endpoint in this condition. So that's very important. We also saw a complete response in 8 out of 15, or 53%. And we saw an overall survival rate of 60% after 2 years, which is really very high in the context of this patient population. And importantly, we also saw no safety concerns. And that's something that holds true for MSC-based therapies in general so far. And that really potentially gives us another major advantage over some of the drugs that are used in these kinds of conditions, which in many cases have a very problematic safety profile. So we're now in a phase 2 trial, which is taking place in the US, Europe, and Australia. We have FDA orphan drug designation for this program, and this trial is at a very advanced stage. We're seeking to enroll 64 patients with newly diagnosed acute graft versus host disease, and we're randomizing them to receive either steroids plus placebo or steroids plus our product, CYP001. We are literally enrolling the last few patients in this trial as we speak, and we're very optimistic of finishing that imminently, hopefully this month. And that would still put us on track to get results from this trial in Q2 next calendar year. So as I said, that's 2 very different trials in very different clinical indications that are both expected to read out within the same quarter. Now, in the interest of time, I won't dwell too much on this. I already mentioned our kidney transplant and diabetic foot ulcer trials, and there's more information about those on our website, as indeed there is about our preclinical programs. And we do have a number of preclinical candidates where we've conducted positive proof of concept studies. Many of those are published as well. And again, that information is all on our website. But the key point for now is that we do have a number of other candidates that are ready to be moved into clinical trials once we have the resources to do that, which we hope to do once we conclude the existing clinical trials. So as I've said a few times, the next sort of 6 to 9 months are absolutely enormous for this company. 2 trials reading out with final results, which are actually each in their own right has the capacity to completely transform this company, and with both of them happening at the same time, it really is an enormously exciting time. I'll just quickly mention that we are generally pursuing a partnering-driven business model, which would involve us entering into commercial partnerships to commercialize these products at the right time. And that may well be once we have results from these 2 trials imminently. We've had a number of discussions ongoing with potential partners, which we're continuing. Needless to say, everybody wants to see the results of these trials, but once those results are in hand, we would certainly be aiming to make some serious progress on that front as well. So I think, Luke, in the interest of time, I can probably pause there. Very happy to take questions and to discuss any issues further that I maybe haven't covered already.
Yeah, beautiful. Thanks so much for that, Kilian. I did have a couple just written down for myself firstly. Now, what I wanted to ask was, from a scientific standpoint, what are the most common misconceptions about MSC therapies that you think deserve a little bit of clarification?
Right. Interesting question. Look, so I think one misconception that we do come up against is that people tend to view MSC therapies as all being the same. And as I mentioned, there have been a lot of trials conducted with MSCs over the last 20 years or so, more than 1,800 of them. But they've all been conducted by lots of different groups, some commercial companies, some hospitals, universities, and so on. They've used MSCs from different types of donor tissue. Some of them are bone marrow, some of them are adipose tissue or placenta, and so on. And they've used different manufacturing processes as well. So there's actually a lot of variability even within all of those types of MSC manufacturing processes. And then Cynata's manufacturing process is completely different. And we've really been building up data to show that makes a difference, that the MSCs produced in these different ways actually have different properties. And of course, in our view, the MSCs made using our platform have very consistent properties, and they show great potency. But I think the misconception is that sometimes people read something about one particular MSC study, and then they kind of make the mistake of assuming that the same issues or outcomes would apply to all MSC therapies, and that really isn't the case.
Interesting. Now, you touched on it before in regards to partnerships, but how is Cynata thinking about strategic partnerships, whether that is pharma or research institutions or even just international groups? Sorry.
Yeah, absolutely. So look, we are a small company, a very small company in the grand scheme of things, and we have therefore really sought to utilize partnerships at every step of the way. And you mentioned their academic partnerships, and we have been very successful in working with academic partners all the way through our journey. So as I mentioned, our osteoarthritis trial is in partnership with an academic group led by Professor David Hunter at the University of Sydney. I'm not sure if I did mention it, but the kidney transplant trial is also in partnership with an academic group, in that case, Leiden University Medical Center in the Netherlands. And most of our preclinical studies have also been in partnership with various academic groups. So we've been very successful in doing that. And I think that's really a win-win situation. We get to sort of benefit from the expertise and infrastructure and so on of those academic groups, and they get to work with very exciting novel therapies like what we're producing. So that's really been what we've done all along. But when it comes to commercial partnerships, our aim really in the long term is that it would be, in most cases at least, commercial partners of ours who would actually be marketing these products. And I think osteoarthritis in global terms would be a good example. There's, as I said, 600 million people or thereabouts with osteoarthritis. If we're successful, there could be absolutely huge demand for this product. This is a treatment that would be prescribed by lots and lots of different doctors around the world. So that's very much sort of big pharma territory. They really have the infrastructure and the know-how to sell products into markets like that. So we would envisage doing a partnership deal with that sort of company to sell that product ultimately. So in terms of when that happens, well, it could well be once we have the results of this trial, which are imminent. Of course, that sort of it isn't certain. Nothing is ever certain until it's all signed. But that would certainly be one possible outcome, and I think a good outcome for us. With some of the other programs, we obviously have the ability to continue the development journey ourselves, at least for a time. We are certainly engaged in partnership discussions about graft versus host disease as well, and that might be one that we seek to partner out. But it is a bit different to osteoarthritis in that it's a much smaller number of patients, smaller number of treatment centers, and so on. So there is more potential for us to take that further forward as well. So we remain open-minded about the exact timing and nature of these partnerships, but we are certainly focused on, in the long term, doing those sorts of deals. And as I said, at least some of them, I think, would be happening in the very near term on the back of positive results in the coming months.
Yeah, okay, beautiful. Now, last one for me before I ask a few questions from the attendees. So, as you do look ahead 12, 24 months, what do you see as the most important inflection points for Cynata beyond clinical trial readouts, whether that's operational, regulatory, or even commercial?
Yeah, so look, certainly one potential inflection point will be those commercial partnerships. So, for example, if we're successful with the osteoarthritis trial and we then go and do a global commercial partnership deal, that would almost certainly involve a very substantial upfront fee, as well as obviously further fees and royalties down the line. That would be something that would be very transformative for the company. So that's certainly the type of inflection point that could come beyond those results readouts. Of course, if we were to decide to take things a bit further, for example, with one of our other indications, then of course there would also be an expectation of further clinical inflection points. But the other thing is with regard to actual commercial approvals. And when it comes to osteoarthritis, in Australia in particular, we have been advised by the Australian TGA, the regulatory authority here in Australia, that this trial should support commercial approval in Australia if the results are good. So we would then expect to be moving forward with seeking commercial approval very soon after the completion of this trial if the results are positive. So that's obviously another huge inflection point. And just to clarify, in the rest of the world, we are expecting that there would be another trial required, but that doesn't necessarily apply to Australia. In fact, the advice we have is that we would be able to move forward with commercial approval on the back of this trial alone in Australia. So that's another absolutely huge inflection point that could be not too far away.
Yeah, beautiful. All right, I might ask a few questions from attendees. The first one there was, given there was no phase two trial for your assessment other than the funding, what gave you confidence to move to the phase three of the osteoarthritis trial?
Yeah, that's a very good question, and maybe I can explain a bit of background for anybody else who's not aware. So we ourselves, Cynata, did not conduct a phase two trial in osteoarthritis and went straight into a phase three, which is quite unusual. But the reason that happened and the reason we were confident to do it is MSCs from other sources have been used quite extensively in clinical trials in osteoarthritis, and that includes trials conducted by the academic group that we're partnered with. So they have used MSCs in sort of phase two type trials, and there's lots of those trials have been conducted by others. Results are published. The results in general point in the right direction, very much so. And so the argument that the University of Sydney had, which of course we got on board with, was we don't really need more phase two trials here. We've already answered the questions that phase two trials are designed to answer. And we think, based on the preclinical data, that Cynata's MSCs have at least similar functionality to the MSCs we've used before, perhaps even better. That remains to be established in clinical trials. But when we know all that, there wouldn't really be much value in doing another phase two trial at this point. And that was then an approach that was obviously bought into by the funding body and specifically agreed with the TGA as well in an advice meeting before the trial started. So yes, it is an unusual situation, but as I said, I think the data that's out there already meant that there really wouldn't have been a lot learned from another phase two trial with MSCs and osteoarthritis, and that meant that what we needed to do was to move straight into a phase three.
Yeah, beautiful. I've probably only got time for a couple more, unfortunately, but there's one here. So, what is the status on manufacturing setup at FCDI?
Yeah, so and again, I'll explain a bit of background there for anybody who's not familiar with that. So FCDI is Fujifilm Cellular Dynamics, which is a manufacturing facility that we're working with. As I did mention, I think we have a strategic manufacturing partnership with Fujifilm. The manufacturing facility that we used initially was a contract manufacturing organization, which is owned by the University of Wisconsin. That was always going to be for initial clinical trials only. It's not a commercial facility. So a few years ago, we announced that we were going to work with Fujifilm to transfer our process there with a view to later stage clinical trials and ultimately commercial manufacture. And so that process is well underway. I mean, we aren't actively manufacturing product for clinical trials at the moment because all of these clinical trials that are going on at the moment, which I went through there, the product was already manufactured for those a little while ago. But our aim is to be using the facility at FCDI to make the product for the next clinical trials. And assuming all goes well in the longer term, that could also then become a commercial manufacturing facility for us. And Fujifilm is very well placed to be that sort of partner for us. So it's we don't have our own in-house manufacturing facilities, so our way of doing this is to partner with specialists in the area, and Fujifilm is certainly one of those.
Yeah, awesome. Last one from myself. Another one from an attendee. with the cash runway for mid financial year 26, do you have plans to be raising any additional capital moving into next year at all? Or
So yeah, we have runway into the second half of next year, which is very important in particular when we have those readouts due before then. So we're certainly not in capital raising mode right now. We don't have existing plans to go and do a capital raise. Obviously, any company has to keep a close eye on its cash position and manage that, but there's no existing plans right now.
yeah, awesome. No worries, Kilian. Well, look, I thank you very much for coming along to and joining a ShareWise webinar. Hopefully, everyone that attended enjoyed and got their question answered. And then we look forward to seeing you at the next one. Thank you.
Great, thank you very much.